Hello, everyone. Thanks so much for being here. My name is Alison Biester. I'm a postdoc at the RCSB Protein Data Bank, and today I'll present a webinar entitled Searching and Downloading Small Molecules in the PDB. I will begin with an introduction to the Protein Data Bank, or PDB, and the Chemical Component Dictionary, or CCD. Then I'll discuss using RCSB PDB chemical search tools, as well as using RCSB PDB search and data APIs. Next, I'll briefly discuss PDB Ligand Validation and downloading CCD data before going into a short demonstration. For this webinar, please submit your questions using the Zoom Q&A function. I'll leave time for questions at the end. Let's begin with an introduction to the PDB and CCD. The Protein Data Bank, or PDB, is the first open access digital data resource in all of biology. It was established in 1971 with just 7 protein structures, and it has grown to become the single global archive for protein, DNA, and RNA experimentally determined structures. The current holdings of the archive exceed 250,000 structures. The archive is managed jointly with our worldwide protein data bank partners in Europe and Asia, plus teams specializing in 3D electron microscopy and NMR. Our rcsb.org website is a one-stop shop for public 3D biostructure data. We offer access to over 250,000 experimental structures alongside more than 1 million computed structure models, or CSMs. This combination ensures we can provide 3D structure information for all proteins constituting the proteomes of humans, model organisms, select pathogens, crop plants, and more. RCSB.org offers several tools for search and visualization. Today, we'll focus on RCSB.org search tools. To deliver consistency and accuracy across the PDB archive, the PDB maintains reference dictionaries for all components making up PDB structures. The Chemical Component Dictionary, or CCD, is an external reference file describing all residue and small molecule components found in PDB entries. Each component is defined by a 3 or 5 character code. Of the over 49,000 molecular definitions in the CCD, most CCD IDs have 3 character IDs, but as of December 2023, all 3 character IDs were consumed. Therefore, all newer chemical components have 5 character IDs. In addition to amino acid and nucleic acid residues, members of the CCD include drugs, enzyme cofactors, substrates, inhibitors, metal ions, and more. Approximately 75% of PDB structures contain non-polymer small molecules. Ligands are common and important parts of PDB structures that are essential for understanding the function of biomacromolecules. In addition to the CCD, the wwPDB maintains a dictionary for biologically interesting molecules called the BIRD. This reference dictionary defines short oligomers, in particular peptides and oligosaccharides. For each entity in the BIRD, there is a molecular definition file, which defines the polymeric sequence, and there's a chemical definition file, which provides a single molecule chemical description. There are approximately 1,200 molecules in the BIRD, and these molecules are present in approximately 4,600 PDB structures. Today, I'll focus on the CCD, but it's important to note when performing chemical searches on rcsb.org that the search will be performed against both the CCD and the BIRD. Next, I'll walk through the anatomy of a CCD file. CCD data are recorded in the MMSIF format, which is organized by MMSIF category and data items under each category. The category shown here is ChemComp, and data items include ID, name, type, and more. Several of the data items here are searchable chemical attributes on rcsb.org, such as name, type, and formula weight. The ChemComp atom category shown here records atomic properties of the chemical component. For the ChemComp atom category, there are often multiple rows of data because most chemical components in the dictionary contain more than one atom. Each row represents one atom, such as O1, C7, O2, etc., corresponding to atoms in the chemical structure. In addition to atom IDs, this category records flags, such as the aromatic flag. As you can see, C1 through C6 are marked as aromatic. This category also records model coordinates, shown here, which are 3D coordinates extracted from the first PDB structure containing the ligand, and ideal coordinates, shown here, which are optimized coordinates. The CCD also has a category called ChemComp bond, which explicitly defines which atoms are bonded and the bond order. For instance, this row defines the bond between O1 and C7, which is a single bond, drawn here. Additionally, the CCD contains computed descriptors, including SMILES and INCHI descriptors, and computed identifiers. The descriptors are key data for chemical searches, as we'll see in more detail soon. So now that we've gone over what the CCD is and the contents of its files, we'll next discuss how to search for small molecules in the CCD using the newly redesigned RCSB PDB chemical search tools. RCSB PDB offers a variety of newly redesigned advanced search tools that allow you to query the database to find structures matching specific search criteria. In addition to structure search tools, RCSB provides chemical search tools, which allow users to query the CCD and the BIRD. RCSB offers two types of chemical search, the chemical attribute search and the chemical similarity search. The chemical attribute search uses structured metadata, such as chemical ID, chemical name, chemical synonyms, and molecular weight. RCSB also incorporates data from external resources, such as PubChem and DrugBank, which are searchable in the chemical attribute search. The chemical similarity search relies on an input chemical structure or formula and searches for molecules that are chemically similar to the input. Here's a snapshot of the chemical search interface on rcsb.org. You can get to the chemical search in two ways, either by navigating to the search dropdown menu on the top bar and clicking on chemical similarity search, or by clicking the chemical search button just under the top search bar. Once in the chemical search interface, you can select the type of search you'd like to perform. This can be a chemical similarity search, a chemical attribute search, or you can combine both tools using Boolean logic. For the chemical similarity search tool, there are three tabs: structure, formula, and chemical component. In the structure tab, there is an embedded chemical drawing editor shown here. The user can begin formulating the search by drawing the chemical structure. In this example, the chemical structure for aspirin is drawn. The user defines atoms, bonds, and stereochemistry, and as the structure is drawn, the descriptor will be automatically computed and written in the chemical descriptor box shown here. The descriptor can be computed in either SMILES or INCHI format, and you can toggle between the two descriptor formats in the interface here. If the user already knows the chemical descriptor they'd like to use for their search, they can paste the descriptor into the box here, and a load button will appear right here. Pressing the load button will populate the chemical drawing corresponding to this descriptor in the chemical drawing editor. The drawing can be further modified if desired, and the descriptor will be automatically updated. The drawing tool serves to improve the user experience while building the search, but it is the descriptor that will be used as the search input. Once the user has defined the search input, they can then define the match type. The user can search for an exact match or for similar ligands, or the user can perform a substructure search, which means that the result must contain the drawn structure as part of its complete structure. Another way to initiate a chemical similarity search is with a CCD or BIRD ID. This option is available under the Chemical Component tab of the Chemical Similarity Search. The user can input the ID here, and the descriptor and chemical drawing editor will be automatically populated. The same options are available as in the Structure tab, such that the user can then modify the chemical structure if desired, and the descriptor will be automatically updated. This tool is a great way to quickly initiate your search if you know a CCD or BIRD ID you'd like to use for your search, but you don't immediately recall the exact descriptor or structure. The Chemical Similarity Search tool also provides an option to use a chemical formula as the search input. This search requires input of element symbols followed by numbers to specify the quantities of each element. Additionally, there's an option to include elements not specified in the formula, shown here. For instance, in the examples shown, the search will return results with exactly 9 carbons, 8 hydrogens, and 4 oxygens, but the results can include any number of nitrogens, sulfurs, etc. For the chemical attribute search, there's an enumerated list of chemical attributes that can be used as search inputs. These attributes include chemical ID, chemical name, and chemical synonym, as well as data that's incorporated from external resources such as PubChem and DrugBank. In this snapshot, I'm also showing a snippet of the search results, which appear below the search query. Once you execute a search on rcsb.org, you will see this gear icon appear next to the text Search API, which is something that allows you to view and edit the search via a Search API query builder that we'll discuss further next. So the RCSB PDB chemical search tools I just described are powerful tools with an intuitive user interface for performing chemical searches on rcsb.org. These searches are powered by APIs, so I'll next discuss the APIs underlying rcsb.org and how you can use these APIs to perform even more powerful searches. Application Programming Interfaces, or APIs, provide programmatic access to data available from rcsb.org. There are 2 main APIs that power the rcsb.org website. In particular, the Data API, which serves to retrieve data when identifiers are known, and these can be PDB IDs or CCD or BIRD IDs. The Search API serves to find which identifiers match a specific set of search conditions. When you browse through the rcsb.org website, you may notice these gears next to the text Data API or Search API, and clicking on the gear icon will bring you to the corresponding Data or Search API query. To highlight how the Data API can be used to retrieve data about chemical components, I'm showing an example here of how to access SMILES and INCHI descriptors given a list of CCD IDs. The RCSB PDB APIs use JSON format queries and provide outputs in JSON format. In the section highlighted here in pink, we have our CCD ID list, and these are chemical identifiers corresponding to aspirin and ibuprofen. In the section below, highlighted in blue, we define which data we would like to retrieve. We can retrieve various data organized in MMSIF categories, including data from the ChemComp category we saw earlier in the presentation. A list of all the chemical component attributes available to query in the Data API is provided at the link listed here to help you build your data API queries. In this particular query, we're requesting retrieval of type, formula weight, name, and formula from the ChemComp category, plus chemical descriptors from the PDBX ChemComp descriptor category. The search API allows the user to find which identifiers match specific search conditions. The types of searches offered are the same as on the rcsb.org website because the API is what powers the searches on rcsb.org. To perform a chemical similarity search in the search API, the user must specify chemical as the search service, highlighted here in red. The key input for the chemical search is the chemical descriptor, as we saw on the website search service. So in this query, the user must specify the type is descriptor, highlighted here in yellow, and must specify the descriptor type as smiles or inchy, highlighted in green. The user must also specify the chemical descriptor itself, highlighted in blue. Then, as in the rcsb.org interface, the user must also specify the match type, that's highlighted here in purple. The match types are equivalent to those on rcsb.org, and the equivalent inputs between the Search API Query Builder and the rcsb.org Chemical Search Interface are listed here in this box. For this search, the match type is specified as graph relaxed stereo, which corresponds to a similar ligand stereo-specific search. Finally, the user must specify the return type is molecular definitions, which means that the result list will be CCD or bird IDs. The search API can also be used to perform a chemical attribute search by specifying text chem as the service, highlighted here in red. As on rcsb.org, the attribute must be specified, that's highlighted here in yellow, and here the attribute is chemical synonym. A list of all chemical search attributes available to query in the search API is provided at the link listed here to help you build your search API queries. The value for the attribute must also be specified, that's highlighted in green, and here we've specified aspirin as the chemical synonym. Again, as with the chemical similarity search, the return type is specified as molecular definition, such that CCD or bird IDs will be returned. I've only scratched the surface of what's possible using the RCSB search and data APIs. If you're interested in learning more, I recommend reviewing the documentation as well as our past training courses specific to APIs. Our team has provided 2 API trainings, one on leveraging RCSB PDB APIs for bioinformatic analyses and machine learning, and another on streamlining access to RCSB PDB APIs with Python. The ability to access RCSB PDB APIs via Python makes these tools all the more powerful as users can perform their searches and analyses more efficiently. To support users' research and analyses, our team at RCSB has developed several scripts for common use cases, some of which take advantage of the Python toolkit for accessing rcsb.org APIs. To learn more about this toolkit, I recommend reviewing the documentation at the link provided here. Available scripts allow users to fetch all chemical component IDs and then fetch the descriptors, fetch and extract ligand coordinate data from PDB archive files, generate mapping files of all chemical component IDs and the corresponding PDB IDs and vice versa, and to search the PDB for the best fitted ligand structure. And this retrieves validation measures that we'll touch upon next. So now that you understand how to use chemical search tools to find your ligands of interest, you may wonder how you can assess specific ligands to determine if they're well modeled. The PDB provides ligand validation on the instance level, meaning that each instance of the ligand in a given structure is independently validated. Doing validation at the instance level is important because the validation measures we use can vary at the instance level. For ligand validation, PDB uses two validation measures, geometry quality and goodness of fit. For the geometry quality validation, the ligand structure is compared against the Cambridge Structural Database, which is maintained by the Cambridge Crystallographic Data Center, or CCD, and that contains high-quality small molecule crystal structures. The validation is done through CCD's program called MOGL. The goodness of fit validation is method dependent because for macromolecular crystallography, the experimental data are diffraction data, whereas for 3D electron microscopy, the experimental data are maps. For macromolecular crystallography, the goodness of fit measures are real space R factor and real space correlation coefficient, whereas for 3D electron microscopy, the goodness of fit measures are atom inclusion and Q-score. The PDB geometry validation is provided in the PDB validation report. Here's an example for aspirin, the component with chemical ID AIN. In this example, we can see in the summary table that there are no bond length outliers, but there is one bond angle outlier highlighted here. The validation report also provides tables for individual outliers, including bond lengths, if applicable, bond angles, torsion angles, and rings. In the individual bond angle outlier table, shown here, we see that the O3, C8, C9 angle should be approximately 111 degrees, but the observed angle is closer to 121 degrees. So the angle is too obtuse, leading to a z-score of over 4. So that's highlighted as an outlier here. The validation report also has a graphical display where the good bonds and angles are marked in green and outliers are highlighted in magenta. For instance, we see the O3, C8, C9 angle is colored in magenta as an outlier. As I mentioned, for macromolecular crystallography, the goodness of fit measures are the real space R factor, or RSR, and the real space correlation coefficient, or RSCC. The real space R factor and real space correlation coefficient are two measures computed per instance comparing the ligands observed and calculated electron densities. RSR measures the difference such that 0 means perfect agreement, whereas RSCC measures the consistency such that 1 means perfect correlation. These measures are provided in wwPDB validation reports and displayed on rcsb.org structure summary and ligand pages. The snapshot here shows the ligand quality slider on the structure summary page for entry 6MQF, which shows the goodness of fit for the best fit ligand in this structure relative to the goodness of fit for all other PDB ligand structures. For 3D electron microscopy, the goodness of fit measures are atom inclusion and Q-score. Atom inclusion measures the fraction of atoms inside the map isosurface at the author-selected contour level. The Q-score calculates per residue the atom resolvability based on the 3D EM map in a manner that is not subject to author-selected contour. The atom inclusion and Q-scores are provided in the wwPDB validation reports, in particular in XML and MMSIF format reports. Again, here I've just provided a very broad overview of ligand validation. So for more detail, I highly recommend reviewing our recent webinar, now available online, called Searching the PDB for High-Quality Ligand-Bound Structures. So once you've found the CCD data you're interested in and assessed the data using ligand validation metrics, you may finally wish to download CCD data. There are several options available for downloading data from the CCD. To download the entire CCD reference file, you can use the wwPDB website, and there's a snapshot shown here on the right. To download individual component definitions, you can use the HTTPS file service. For batch ligand download, you can use the service available on the rcsb.org website, linked here. Through these download services, you can access MMSIF, SDF, MOL, and MOL2 file formats. Additionally, you can download descriptors for all ligands in the CCD using the download links on the wwPDB website. Now, we'll move into the demonstration portion of the webinar. So in this demonstration, I'll briefly demonstrate how to use RCSB PDB search tools on the website and via API query. Then I'll show how to find ligand validation information on the rcsb.org website. And finally, I'll demonstrate a batch ligand download on rcsb.org. Okay, so as I mentioned, to navigate to the chemical search service, you can go to the search drop-down menu on the top bar and click on chemical similarity search. This brings us to the chemical search service where we have access to chemical similarity and chemical attribute searches. Let's begin with a chemical attribute search. When we click on chemical attributes, we now have access to this search bar where we can either type to find an attribute. For instance, we could type drug to see attributes containing the word drug. Or we can browse the drop-down menu, which has a full list of enumerated attributes that we can use for our search. So let's say for this search, we'd first like to find drugs that are approved. To do so, we can click Drug is approved and select the value yes. We can see this search returned 855 molecular definitions. We may want to narrow our search down further, and we can do that by adding additional search attributes. So the chemical attribute search supports full Boolean logic, so we can add another attribute to include another search criterion, and we can use Boolean logic with either an AND logical operator or an OR logical operator. So let's say we would like to only include chemical components that were released prior to the year 2000. We could do that by clicking on chemical component release date and then specifying the date as before 2000 on January the 1st. Then we can hit search here. And so now we only have results that meet both of these search criteria. And we can see we've narrowed down our search substantially to 170 molecular definitions. This is still a pretty large search to work through. So another way we can narrow down our search is by adding a chemical similarity search using AND Boolean logic. The advantage of chemical similarity search is it allows you to define specific chemical groups that you'd like to see in your search results. So to add this search, we just click on chemical similarity. And so this is now added and combined using AND Boolean logic. So let's say for this search, we'd like to narrow down to only components that contain a phenyl moiety, meaning they have a benzene ring with an alcohol or OH group attached. Now we have access to the chemical drawing editor, so we can begin drawing. And we can see as we start drawing this structure, the chemical descriptor box is automatically populated. and it's updated as we update our drawing. So now we have our phenol drawn, and we can toggle here if desired between SMILES and INCHI format descriptors. In this case, either descriptor format is appropriate, but there may be some cases where you prefer one descriptor type over the other. For instance, when I'm performing searches for a component that contains a metal, I favor a SMILES format descriptor because the INCHI format descriptor doesn't encode information for metal coordination bonds. In this case, either descriptor format could be appropriate. We then need to define our match type. So if we'd like to return results that contain the phenol as part of the overall chemical structure, we need to change the match type to substructure. Since this molecule doesn't have a stereocenter, it doesn't matter if we do stereospecific or including stereoisomer search. We can choose either. Now we click the search button to execute. And as a reminder, we can see that this search must match all of these search attributes because we have combined the search criteria using and Boolean logic. So this will narrow down to a smaller selection of 32 molecular definitions, all of which contain this phenol. As you can see, if we scroll through some of the search results. Before we look more closely at one of these results, I want to draw our attention to the search API, which we can find by scrolling to the top of the page. So we see here the gear icon next to the text search API, and we can click to see the search API query in the query editor. I want to draw your attention to a couple of things in this query. In particular, we can see that our search criteria are combined using the logical operator and. So this narrows down our search to something that matches all of our search criteria. We can also see in this query that we're invoking the chemical attribute search service based on the specification of text chem to search for approved drugs here. And we're also invoking the chemical attribute service to narrow down the release date of the chemical component right here. Finally, I want to draw our attention here to where we're invoking the chemical similarity search service based on the specification of chemical. We specify as well the type as descriptor and the descriptor type as SMILES, and the match type as substruct graph relaxed stereo, meaning it's a substructure stereo-specific search. You can execute this query by clicking here on Execute Query. And as this search completes, we will see the same set of results as we saw on the rcsb.org website, and that's because it is the same search. This is the API that's powering the search on rcsb.org. Even so, it is very useful to learn more about using the API query editor because it will allow you to perform even more powerful searches. So we can see, as expected, there are 32 results returned. And if you were to check, you would see that the identifiers match with the identifiers that we saw returned in the previous search. Now I'll take us back to the search results on the website, and we'll investigate one of the results a little bit more closely. So we can see we have a number of different results here. And for the purposes of this demonstration, I'm going to select this component, TYL, which is Tylenol, otherwise known as paracetamol or acetaminophen. So to click in, I'm going to open this result. And we're taken now to the ligand summary page for TYL. We can see that there's a 2D depiction of the ligand here. as well as the 3D coordinates depicted here. We also have access on the summary page to the chemical component summary, which shows the name, formula, and more, as well as chemical descriptors in this box, and additional integrated information from Drug Bank, shown here. In order to access validation information about this ligand, TYL, we need to look at this ligand within specific structures because the ligands are validated at the instance level within individual structures. So in order to search for structures containing TYL, we can click on this link here to execute the search. And this search returns 10 structures. Let's sort the results so that the newest structures are shown first. And for this demonstration, I'm going to click into this structure for CUT to investigate further. Now I've brought us to the structure summary page, and we can see on this page the ligand quality slider, which shows the goodness of fit for the best fit TYL in the structure as compared to the goodness of fit for all other PDB ligand structures ranked. And so we can see in this case, it has a relatively good goodness of fit. And to see more details, we can click on the vertical bar here. So now we're brought to the ligand page on which we see these 3 2D plots that show the goodness of fit on the x-axis and the geometry on the y-axis. In the left-hand plot, we see the best fit instance in this entry compared with other instances in the entry if applicable. In the middle plot, we see the best fit instance in this entry as compared with best fit PDB instances with the same target. And then in the right-hand plot, we see the best fit instance in this entry as compared with the best fit PDB instances with a different target. Additionally, at the bottom of the page, we can see these data in tabular format. So one thing that's interesting here is that although we saw the goodness of fit is quite good for TYL within this structure for CUT, there's actually a better fit ligand with better geometry in a different structure. And that is in entry 5PBE. In this bottom table, we can actually navigate through this link to see the atomic coordinates of this ligand overlaid with the experimental density. And whenever you're performing structural assessment, I highly recommend, in addition to viewing the validation metrics, you also view the coordinates and the experimental density. So let's go ahead and open this up. And we can see the coordinates for this TYL ligand along with the experimental density. And we can see it is quite a good fit. And by visual inspection, we don't see obvious geometric distortions. Even so, I always recommend viewing the validation report to see if there may be individual outliers that are not obvious by eye. You can access the validation report through this Download Files menu, and you have access here to the validation report in PDF, XML, and CIF formats. Finally, I'm going to take us back to our search results and show how we can perform a batch ligand download on rcsb.org. So if we scroll up, we can see we have the option to download multiple files from our search results. And by default, all is selected, but if desired, we can change to only download selected files and select whichever are the files of interest. For this demonstration, I'll leave on the default of all, and we can generate our list of IDs to download by clicking on this Download All/Selected Files button here. Another way to navigate to the batch ligand download is through the download drop-down menu on the top bar. So from this menu, you can click on ligands, and this will take you to the same batch download page where you can input your IDs of interest. So now we've generated the list of IDs for download, as you can see here, and you can modify this list if desired. For instance, you could add ATP to the download list if you'd like. You can toggle the content type as well as the file type here. And once you're pleased with your selections, you can click Generate File Batches for Download. And then in order to perform the download, you just click on the batch, choose the directory for the download and hit Save. And then you'll have these files downloaded onto your computer. Okay, with that, I'm going to take us back to the slides for conclusions. So in this webinar, we saw that the redesigned RCSB.org chemical search offers intuitive chemical attribute and chemical similarity search tools. Also, the RCSB PDB APIs enable even more powerful search capabilities. We also saw validation metrics provide key insights for finding the best modeled instance of a given ligand. We also offer diverse download options to allow download of one, many, or all components in the CCD. Last but certainly not least, I'd like to thank the whole team at RCSB for building all of these tools to help support the community. And I'd also like to thank our funding agencies, in particular NSF, NIH, and DOE. And with that, I would be happy to take any questions.